closing remarks. I appreciate everyone's questions and participation in this year's symposium. It's been a joy to run and to host everyone. So yes, thank you all. Our final keynote speaker, last but not least, is leading a significant new chapter in Texas viticulture. In January of last year, Dr. Svyantek established the Texas A&M grapevine breeding program within the horticulture department. His research is focused on making breeding more efficient and affordable by increasing selection accuracy and reducing timelines. The background that he has from Auburn and North Dakota State University has helped to develop his mission to develop new cultivars specifically tailored to the unique needs of regional farmers. And finally, to share his vision of the future of multidimensional breeding, please welcome Dr. Andrej Svyantek.
All right, so my name is Andrej Svyantek, and we're going to talk about how we're establishing a horticultural breeding program from scratch. And so to put things in perspective, today's talk I'm gearing towards our students, to faculty who may find themselves in my position someday starting a breeding program from scratch without germplasm, without lab material, and without field material, and as well as to myself to look back in the future at what have I done? Have I stayed on the path that I'm trying to target? Horticultural breeding and horticultural crop production in general is a very long-term investment. We have investment upfront in which we're taking years and years to collect data and make decisions and make populations that we're pushing forward. And so this requires a team approach to pass through the years. And when we look at the horticultural needs in Texas, we have a lot of diseases, but we also have a lot of abiotic stress, but we also have no water, but we also have hot summers, but we also have turbulent winter events. And so the needs are expansive and demand us to tap into all of the diversity of our species. And in my case, we'll be talking about grapevines. Beyond that, in grapevines and in perennial crops and in any of our crops, we have a number of obstacles to our observation of an actual plant's value, which leads to phenotypic variance that we have to overcome. And some of the ways that we can overcome that are by developing selection methods, screening platforms, and different techniques to try to winnow down as much germplasm as possible in as short a period of time. And so these will be the main points that we'll keep bouncing back to throughout the talk. And starting things out, it's really important to look at breeding as a team effort across decades, across generations. And in the case of grapevines, we have this massive investment that we're putting in any of our horticultural crops that take years before we have selectable phenotypes. So we're spending money on greenhouses to start the plants. We're spending money on trellis, on landscape to establish and train them for years until we have salvageable phenotype that we can talk about, select upon, or publish. And so the worst part of this is after we decide we don't like a plant, we still have expenses associated. We can't till in vines. We have to rip them out by hand. We have to put in expenses on trellis and site rehabilitation so that we can continue to utilize our land for selection. And so we're investing on plants that we don't know whether they're worthwhile, and then we're investing again when we decide that they're not worthwhile. And these are contributors to our expenses in our breeding efforts. And the other thing about our breeding program and horticultural crops is that many of our perennial crops have long extended juvenile periods. So in the case of grapevines, we're looking at 2 to 5 years before we have fruit to phenotype and to push a vine forward in a yes or no selection method. And then as it starts to carry on downstream, it's, the fastest case is 10 years. Realistically, most breeding programs are 25 to 30 years between cross and cultivar release. And that means that we're looking at 40 to 50 years before the actual industry starts to adapt and evolve and utilize the breeding program that might have been, breeding material that might have been developed long ago in the past. And so we have a lot of students who are working incredibly hard, and if they come to my program, they're going to develop seeds, develop beautiful crosses, and they might never see fruit from their efforts. And so it's really important for us to understand and develop a generational mindset in this breeding program that we're doing this not for ourselves, but for the future. And a lot of the material and sort of sites that I'll be talking about for my breeding experience is going to come from beautiful and balmy Fargo, North Dakota, where we had a number of different seedlings that we planted out a long time ago and have had generations of students come through phenotyping them to get us to the point where we are now. So by the time that I started my PhD, people like Dr. John Stanger, Harleen Hatterman-Valenti, and Colin Otwater had already put in the work to develop crosses. We germinated something like 17,000 seedlings and eventually planted 4,500 in 2017. And of those 4,500, about 1,000 were genotyped to any significant degree and utilized in students' projects. And there were 4, 5, 6 years before we had fruit to characterize, before we had meaningful projects for students. And so a lot of us on that picture, we spent our time on our hands and our knees, pulling weeds, taping vines, and now we're starting to see fruit from that labor. So breeders in horticultural crops and in any crop, we have to work in the past, the present, and the future. We're pulling from the past in the germplasm that we're utilizing. We're actively making selections in the present, and we're guiding this towards a long-term future and trajectory. And so it's important to understand the needs of our growers and the needs of our landscape. So if we look at Texas as a whole, I will reiterate how fun it is going to be to breed for grapes in Texas because of all the challenges that we have and the diverse climates that we're situated to fight against. So our breeding program is stationed here in College Station. We have a Hort Trek site just down the road. But if we're breeding for grapevines that need to be deployed in Denison, North Texas, in the Texoma AVA, down in South Texas, or in Hill Country and High Plains, we have a number of different environmental constraints that lead to different disease and other parameters that we need to preemptively select for in our breeding efforts. So whether it's the timing and the rate of rainfall and the impact on our fungus, or the fact that some of our locations may not even get chill hours or lose their leaves before their second spring comes the following year. We need to tap into the diversity of grapevines across the world, and more than anything, the diversity of grapevines from here in Texas. So this is from iNaturalist, not unlike you. We pulled data sets. My student, Ashmita Bhushal, is working to identify which flowers will bloom when, so she can identify native cut flower potential options. And so the iNaturalist data set gives you the date of an observation, and she's working hard to understand more about that. We did the same thing with grapes, and we can see and identify different accessions and locations where there might be an opportunity for us to collect and capture the diversity of Vitis in Texas before it's too late. And the diversity of Vitis in Texas has been incredibly important to the state history of Texas and its development. So from the early days of the Texas frontier, Gideon Lincecum characterizing species, speciation, and diversity of plants and animals, and even some of our leafcutter ants across Texas. This was some of the foundations of understanding what it is to have organisms that can survive in the state of Texas. And following him, T.V. Munson, from the 1870s to the 1913, he was a private breeder running a nursery operation in North Texas in a place called Denison. And he was instrumental to not only developing grapes that were tolerant for Texas, were instrumental in my my master's introduction into grapevines, but also in preventing and overcoming some of the threats that were an obstacle to the entire grape and wine industry in Europe. So through his assessments of diversity, finding grapevines that were tolerant in Texas, were tolerant of calcareous soils in particular, and were also resistant to phylloxera, he was able to distribute vines, and through the act of grafting, overcome the challenges, and was one of many Americans who tapped into the species diversity of America to help save the global grape and wine industry. Beyond grape diversity, grape production has been foundational to our department of Texas horticulture, and we're really, really proud to be part of a department that's literally written a book on growing grapes in Texas and the Southwest. And as our colleagues have laid the foundation, our industry has grown. So the grape and wine industry in Texas is punching above its weight class. About 10 years ago, we wouldn't have been in the 20s. We might have been getting close to the 20s or teens as far as acres planted, and we've rapidly grown to become the 5th largest state based on acreage. Despite that, we're the 2nd largest state based on economic impact. And we have a bright future. But we do have continued obstacles to our success. Not only do we have the pests and pathogens that we could rattle off their names endlessly, but we have interesting environmental constraints for growing grapes here in Texas. So I'm really, really fortunate and lucky that I got to start my career here. About 1 year, 1 month, 18 days, and 8.5 some odd hours ago, I started breeding grapes in Texas. And we've been assembling our team with our postdoctoral research scientists, Venkateswara, with our graduate students, and with undergraduates who are willing to put in the work. And we're really fortunate because despite all of our challenges, no one in the world has it better than us. We're the youngest public grapevine breeding program in North America, and I'll continue to say possibly the world until somebody tells me otherwise. And in light of certain aspects of alcohol sales nationally, internationally, this may remain the same statement for a long time. But we're serving, here in Texas, the single largest geographic area. And we're at a really, really unique environment in which we can tap into diversity from the southeastern United States, southwestern United States, and the Great Plains, and we are positioned to serve those growers as well from vines that we identify that are adapted for our conditions. The folks who have been leading the charge in developing our grape and wine industry are I'm really fortunate to call colleagues and friends and coworkers. So we have a core group of viticulture extension specialists across the state and researchers who are all working to serve the grape and wine industry. And I'm really, really excited to be part of 3 new hires. We had a faculty cohort hire that brought 3 of us in simultaneously to work to address the problems of our growers, whether it be physiology with Lorenzo Rossi or secondary metabolites with Ahmed Darvish. We're positioned together to collaborate, to work together, and to cooperatively address the needs of our stakeholders. And coming to Texas, Texas is a large state. We've also been fortunate to hop on the bus and visit some of our different research and extension centers to understand more about the needs of Texas and our resources and our collaborators. And so we got to see places like Lubbock, Stevensville, and there's Hope at Halfway There Research Station, and we're about halfway there in our understanding of what we need. We're starting to connect with different faculty across the state, across the different capacities to serve, because we're new faculty and we all understand that we need to continue to grow to better serve our farmers. We need to continue to grow our collaborative network, continue to grow our own internal teams, and most importantly for our internal focuses, continue to grow out our tools and our data streams so that we can most appropriately address the problems at hand. Some of the biggest problems at hand in the case of our crops or breeding are the intrusion of non-genetic variants. So grapevines are this really finicky crop that they need everything, and they, if you do one thing wrong, they might die. And so grapevines in their life cycle, we can't pull them, we can't choose our planting date but once. And so our vines need to be durable to survive winter. They need to bloom at appropriate times for proper pollination and not to get frosted. And they need to ripen at appropriate diurnal temperature regimes to have retention of sugars and acids in appropriate quantities to achieve product quality for our entire product goal pipeline of making high quality fruits and beverages. And anything that we do along the way has an impact. So one case example would be if we remove a single leaf in the fruit zone or multiple, we have this butterfly-like effect on our grapevine's physiology, capacity to survive winter, capacity to fruit. So fruit zone leaf removal is a common management practice utilized by growers in which if you extract leaves, you open up the canopy, you end up altering your harvest dates, you end up altering your ripening chemistries, your ripening parameters because of the interplay between the environment, between the sources and the sinks. And so we have canopy management practices that have these lasting impacts on our in-year chemistries and our in-year disease, and also longer-term impacts on our cane hardiness, our cane survival, our carbohydrates in our wood that overwinters, or the floral differentiation that occurred in year 1 and dictates our yield in year 2. Because of all these interactions ongoing at all time points, We have a field of management for grapevines specifically called horticulture. And horticulture is the foundation of many of our understandings of agricultural challenges, whether it be plant pathology, soils, horticulture, or food science. Viticulture as a component of horticulture stands as one of the areas in which humans have committed a large amount of efforts to understand plants' responses so that they can make maybe more sugar, better alcohol in the end game. So one of the examples would be, say, the development of specific fungicides like Bordeaux mixture, initially used to just keep people from eating grapes. They eventually found out that it kept some fungus from attacking as well. The earlier mention of our overcoming phylloxera through the combination of a scion and a rootstock, combining the best of both worlds so that we could survive in our soil constraints and the pests that might be there. We also have efforts on physiological innovations like root zone deficit irrigation, and all of our wine science is based, and microbiology and food science is built on our understanding and desire for a stable and clean glass of wine. And here today we're talking about what we're trying to do in the realm of plant breeding. So keeping this in mind, grapevines are finicky, they are responsive, they are needy plants, and we're trying to understand what they need or what might be in their way. So we're trying to start to map the spatial and temporal variation that might be an obstacle to our vines, and to understand the landscape that we're going to be entering our grapevines into. And Texas is really weird. In our driest year, our wettest locations are drier than our driest locations in the standard year, and vice versa. So we can have disease pressure in the high plains that's comparable to disease pressure in East Texas. We can have a humid condition anywhere because we get a world of different weather here in Texas, and the years change, the days change. Everything is out to get us if we're growing grapevines. And so we're trying to characterize and understand who our obstacles are so we can attack them preemptively. Our obstacles include things like Pierce's disease, which is widespread and also has no cold curative factors. We have variation in our growing season. We have variation in our soil pH conditions and in our soil texture class that all play a role in how we're going to choose different rootstocks, different scions, and what sort of genomic background or diversity we're going to utilize to try to deploy in those environments. And it's really important because a vineyard manager has all of these decisions that they're trying to make. And if they make a mistake, they lose their crop, they eventually could lose their farm, and eventually we end up with communities that are essentially without folks and without farmers. And so we need to preemptively make it harder for our farmers to make a mistake, because things change and drought years come and diseases come. We need to make durable, lower input grapevines that are capable of still producing acceptable quality and yield in our growers' different environmental and management contexts. And that's where we come to some of the different ways that we've been trying to make selection for these purposes. So we'll be talking about a couple different things. First, we'll talk about powdery mildew again, and then we'll talk about a tiny bit on downy. We'll talk about fruit chemistry, and not so much how it translates to wine, but how we can characterize it a little faster, a little better. And then we'll spend a little bit of time on cold hardiness, because I guess I just miss winter. So when it comes to powdery and downy, on the left, powdery goes on the surface, downy goes on the bottom of our leaves, and they both will destroy an entire crop. Powdery mildew, if it gets early enough on the vines, you won't have to harvest or be tricked into harvesting, but if it comes a little later in the season and you still continue to decide to pick, you'll end up with a wine that tastes like hints of shoe, mushroom, and some other things from a gym locker room, versus downy mildew will sort of clip off your entire crop before it gets there. So we're really excited to be working in collaboration with folks from the Grape Genetics Research Unit and Geneva, New York, who have developed a technique with this tool they call the Blackbird. It takes images frequently of leaf discs that have been inoculated, and with high resolution and human effort to train a neural network. It's capable of annotating the growth and sporulation and hyphae development of these pathogens across time. And so these are what they look like real up close on Thomson seedless, the susceptible check at different days past infection. And it's really exciting. So what we did this past summer was we submitted some seedlings to them, and we utilized this information to make some selections and to make some decisions on destroying individuals. But we also submitted most of our Munson cultivar sort of germplasm repository that we're using as parents from TV Munson, who was breeding very, very long time ago in North Texas. And we were able to find that we have potential parents with high rates of resistance to the diseases, not only across multiple isolates, but also across an extended period of time. And none of this would be possible without somebody besides a human taking the measurements. When it comes to fruit chemistry and wine, this is where things start to get pretty tricky. So Brix, pH, and TA, that's what we utilize to characterize any basic fruit parameters. Your Brix goes into how much it's your sugar content and translates into your ethanol. Your pH and your titratable acidity translate into some of your capacity to maintain a stable wine, and they're both reflections of the same sort of thing of your ripeness. Beyond that, you can get into your aromatic compounds, your phenolic compounds, or your specific acids and sugars. And the tricky thing about fruit chemistry is that it changes every single day. It changes based on the time of day and also the day and the gradual development of the vine. And we have to sample live, and we have to process pretty much live for the most part due to some of the interactions of our compounds with cold. And so sugar development for our breeding lines, we can range from just under 5 to almost 25 at a given sampling event. And we get this broad spectrum of what's going on in a wide germplasm pool or even in a narrow cross. And so we need to be selecting more frequently. These are from about a 10-day interval, and you can see how the mean moves up from about 13 on harvest 1 in 2020 to harvest 2 in 2020, about 17, and then on and forward. And so we're missing a lot of meaningful information even at this 10-day sampling interval, but it's the best that we can do. It gives us a spectrum of the basic chemistry that's going to translate to fruit wine quality. And so this is from Venkat's dissertation, and he just recently published it. And what he found was that there's a lot of different factors controlling chemistry. It's a quantitative trait. It's to be expected, but there's so many factors and they're biologically relevant, but there's not a direct pathway for us to utilize this in selection. And so he's been working on developing different genomic prediction methods to see if we can figure out and solve for seedling characteristics prior to deploying seedlings into the field. But a lot of this is limited by the amount of information we have. So that data was from 9 sampling events across 3 years, and you'd think that that would be enough, but we need way more. We need more sampling, and we need to do it in a less time-consuming manner and gain more return and more data points from that investment, because our time spent on phenotyping is expensive and we can't get it back, and it happens all at once for all of our different crops in many cases. So one of the things that we're really excited to start to work with, thanks to some funding from A&M AgriLife, is utilizing NIR screening for some of our fruit crops to non-destructively analyze and predict ripening across time, and then also to pair that with FTR characterization of the liquids so that we can expand the trait set that we're starting to look at, whether it be the specific sugars and acids, the YAN, which goes into how well a wine can ferment, the phenolics or other things, and start to feed that back into other data pipelines so that we can develop genomic prediction that's responsive to our time and our day of sampling and the influence of growing degree days into that. In general and in whole, all of us are here to breed to serve farmers and families, and that means that we in whole have to be working on everything everywhere all at once, and grapevines are the epitome of this. We have a concept called terroir, in which the landscape that you grow the vine, the person cultivating the vine and their management decisions, and the specific cultivar being grown lead to the impact on your final wine quality. And this concept means that there's a lot more than the cultivar influencing what's going on in a single sip of wine. So we have an animation. You can look away. It's not as smooth as I wish, but this is sort of a perspective of what's going on in a single glass of wine. So we have interactions that are going on between our scions, our rootstock, and our pest pressure. We also have interactions of genotype by environment by management when we come to our wine fermentation, whether we're adding oxygen, whether we're agitating, what our temperatures of fermentation are. And then we have this management level on our sensor who's tasting the wine. What was the light like? What was the temperature like? What did you eat before? Did you drink before? How many wines can you taste? And so just like us now here, how many talks can you take? We're almost there, we're almost done, but we gotta finish this one last glass. So, what I'm saying is that there's a level of complexity in any one of our wine glasses that is oftentimes not related to the genotype, oftentimes not even related to the management in the vineyard. You can make bad wine with good grapes, you can't make great wine with bad grapes, all these different sayings, but you need everything to be in place to get to the final product in a meaningful fashion. And our sensory data approaches require somebody to donate their tongue to science and then some, so that we have some sort of form of consistent approach. When we look beyond whether we're making fruit or making wine, in the whole, grapevines are an incredibly expensive crop. They have a high price per plant, we have a high price per phenotype, and our phenotypes are generally variable. So we need more and more phenotypes so that we can resolve and understand what's actually happening in a given grapevine. And so I'll be talking about that next as we're trying to work towards some level of capacity for on-farm predictions so that we can project what a genotype might be doing where based on spatial or temporal data. And we're talking about that in the case of cold hardiness. So cold hardiness is really important for grapevines. We can't dig them up. We can't cover them in a cost-effective manner. We cannot. They're stuck in the ground once it gets cold. And for my PhD, cold hardiness was incredibly relevant in North Dakota, a location where we'd sometimes have less than 4 months and rarely more than 5 months of a growing season. And the rest of the time it was a tundra. It was colder than the North Pole at many times of my life in North Dakota. And so it was important that a grapevine would be adapted to cold hardiness. Colder than Mars. And when you get colder than Mars, you have grapevines that will literally explode. And if they don't explode, they'll collapse mid-season because they don't have anything in their phloem connecting themselves. But as important as it is in North Dakota, it's equally important here in Texas. So we had Winter Storm Uri in 2021, and that wiped out a lot of the South Oklahoma growers that I was working with, central Oklahoma growers, but it also damaged a lot of the growers here in Texas in the Gulf Coast region where vines like Blanc du Bois, unfortunately, exploded. So what happens when we're trying to survive winter? Our dormancy period is a hidden phenotype that we don't oftentimes think about, but hardiness fluctuates with our environmental cues. We get colder, we acclimate, our vines get a little more resistant in a lot of different ways, but anytime that our cold temperatures overcome our lethal temperatures, we end up having a really bad time as growers, as farmers, and as researchers. When we're trying to phenotype for cold hardiness, the gold standard would be selection winter, a killing event that comprehensively killed across the board. But we have to remember that death comes differently to young vines, to old vines, to different tissues. And so even a killing winter, if you could generate it, it's not a reliable phenotype that you can always select on unless you have everything the same at all times. So one of the techniques that we use in place of calling in a winter storm that freezes all of our eyes shut is we utilize freeze chambers to assess death of canes, electrolytic leakage, and or differential thermal analysis as a response that we can select upon. So freeze chambers are challenging because you have to put people into the field collecting wood at a time when nobody wants to be in the field. And it takes a lot of effort and a lot of dedication. That's Bulent Kosa, and I spent many a day driving in a truck with him out to the middle of Absaraka where we would get our truck stuck and we would just try to stay warm enough so that the battery wouldn't die. What we do with canes when we bring them back is we turn them into these little burritos that we put into a freeze chamber. We place the burritos of different treatments of different genotypes into a freeze chamber, and we need a lot of canes to get resolution as we're computing the LT 10, 50, and 90 from their death. And then we also need a lot of time and consistent observations of the canes as we pull them out of the freezer, because an individual needs to cut open and examine whether or not phloem, primary, secondary, and tertiary buds have been damaged by the temperature they've been exposed to. The only way to do that is to slowly, manually ramp down the freezer, pull out samples every -5 or so degrees, ramp down. So this can put people in the lab with a freezer in the middle of winter well into the next morning. The alternative to this is one that we've been utilizing more extensively, and that's differential thermal analysis. It's still got the limitation of a freezer, but it has the capacity for observations without a human. So we collect, in this case, we are limited by the number of individuals we can put into a freezer at any given time, and the time that it takes from the freezer start to finish to preparation for the next freezer run. So it takes us about 2 days for any given sample. And that can become hard and disjunct when we're trying to combine across freezing events or across different dates. And the other challenge of only 54 genotypes is that sometimes they're dead when you put them in, so you end up with somewhere like 40, 45 on a good sampling event of calls from this trait. But we freeze them, we ramp them down just the same, so it goes from like, let's imagine there's a number and it's room temperature, and then we get to -45. It does that every hour, dropping about 5 degrees. And then we see this signature of screaming of buds as they release the latent heat of freezing, and that's used to annotate which buds died at what temperature. This takes a lot of work, and so we're trying to look into new approaches for selection that can integrate this with genomics or for recommendation for growers. And there's a really exciting model that was published about 2 years ago that we can use as maybe something to jump on top of. So this is the Vine Cold database. It was published by Hongray Wong and Jason Londo, and they took DTA data from collaborators in Texas. You can't see Texas, but that's Texas down here. And Texas, New York, Wisconsin, Washington State, all these different people who were always working to solve growers' challenges with grapes and trying to predict whether they had lost their crop. And feeding this cold hardiness data in conjunction with winter actual temperatures, their extremes, the chill units, into multiplexed machine learning, auto gluon, random forest, Excel something, there's a cat one as well. Those all together chew through this data set and create this really useful tool for our growers where they can integrate and actually select a location and look at both real-time as well as historical trait predictions of different genotypes at their environment in sort of like a synthetic data set based on the modeled hardiness. And so this is cool. A grower or a researcher can extract the 54 cultivar responses at a site-specific location, and this gives a lot of intel if you're growing one of those 54 cultivars, but it doesn't necessarily help us as breeders. So we're trying to figure out if we can leverage this pre-existing data to strengthen our own predictions. So this is my DTA curve. It's really beautiful, and it took us collecting Vitis riparia Clint in the middle of winter from November to about April. Every 2 weeks, we would trudge out into the snow to collect these canes. And if we can combine this sort of to weight our means with the data from the other modeled individuals, we're able to get a much more intuitive curve that matches what's being shown from a limited data set. And so we can turn our general sets of many different varieties into from a sort of squiggly, scraggly curve into something that approaches potentially realistic physiological responses to the cold triggers. And then we can come back and potentially start to utilize that in recommendations or in interrogation for our growers on a landscape map so that they can understand the site-specific challenges that they might be facing with cold from varieties that were never tested as part of the initial model, but we can hitch or ride on that model potentially for our own purposes. And so that was done with about 10 observations, but we're trying to see, this is the work of Ramesh Pili. He had 2 of his classmates who were here. We're trying to see with some of this data if we can do the same with our breeding program material, where we have, that's 400, 500 observations in one of those clusters. And it looks something like this, but we have trouble combining them because of the variation between our experimental freezer runs, between the day-to-day variation of the plants, and because all of the controls that we included, only one of them was able to give us 2 data sets. So when we have our control varieties dying and we need to solve without them, we can generally combine across those sampling events and try to get something approaching a realistic curve. Or what we're trying now with Ramesh's data is to hitch a ride on some of the modeled data to see if we can create a combined data set that's going to allow us to fill in and wait and follow the predictions of the 54 cultivars at any given site. And so we can take this is 400 individuals, he's got data sets with 600 individuals. We can start to create a larger, more interpolated, more predicted data set that can be analyzed, and maybe we'll find that this is not useful, but maybe if we start to expand and go beyond this into other traits, we'll get to the point where we can give our growers this day-to-day specific, site-specific map for their station, for their farm, of traits not only cold hardiness, but also their yield, their parameters of disease, whether they should be thinking about a preventative or a kickback fungicide application based on disease pressures and based on their vine's inherent susceptibility. So winter isn't the only thing that we need to survive. There's a lot of other phenotypes that we need to be actively selecting for as we try to deliver varieties to our growers that are resistant to our stresses and are also capable of surviving here in Texas. And if we can do that, we can start to build out our own open data sets, start to tap into and extract values from historic data that we can repurpose to strengthen our prediction capacity. And that, I think, is going to give us an opportunity to make heritable change in the face of our enemy, which is error. So these are some tools, phenomics, genomics, modeling. They're going to help along the way, but nothing's going to replace me, hopefully. We need boots on the ground. We need people who are willing to train vines and sweat and work. And so now we have one more slide. It's visually jarring. This one I put in directly for you. So everything, everywhere, all at once. It's a little overwhelming. There's a lot going on. There's all these things that we're trying to work towards and breed to. But if we can focus and we can isolate something to focus on and work towards, we as students, we as people, we as researchers can make an impact that's a little more lasting and less explosive. So putting things back in perspective for our students and for researchers who might find themselves in positions similar to me, starting from scratch and trying to figure out the landscape. For students, I think the most important thing that you can do is collect more data and collect more data. And then after you've collected more data, do it one more time, but digitize it and annotate it. Annotate how you did your ratings, how you did your methods, how you measured. What did you consider the widest point of the trunk, and how did you determine it, so that someday we can start to utilize your data sets to leverage so that somebody won't have to take the same tedious measurements that you might find yourself doing today. And for breeders who might find themselves in my position, there's importance of sort of targeting a few traits and making sure that you can have achievable timelines and progress that will be rewarding to your career, because the career is going to take a lot of time and effort. And more importantly, it's going to take a team to get over the final edge in any form of breeding. So when we take in everything in the state of Texas, the most unifying force that's fighting our grapevines is Pierce's disease. It's a pressure in which we have no curative action. As we spread farther north with this bacteria, we lose our capacity to just avoid it by planting where it's not been introduced or gets killed by cold. And so our breeding program in general is focused on developing Pierce's disease-resistant grapevines that, through our collaborations with folks like Lorenzo, Ahmed, are able to overcome stresses, are able to utilize proper secondary metabolites to fend off and ward off pathogens, all the way towards us trying to develop world-class table grapes with high wine quality. These are the points. Things take time. Things take a team. You need to utilize everything that's possible in the species diversity of your species to potentially overcome the challenges, and nowhere gives us the same suite of challenges as we do here in Texas. So we need to utilize everything that evolution has awarded us. We need to improve our processes of phenotyping and the way that we can collect data and retain it and communicate it to the next student who follows behind you. All so that we can start to make more accurate predictions, projections, and selections of grapevines or your crop. And so it's a lot to take in. It's a lot to work towards. We're breeders, and we need to know why we're here and what we're doing. So you need to have a really good understanding of your organism, who you're growing in it, and so you can anticipate the sources of variation in any metric that you're taking. You need to work in the worst conditions in the world because growers are relying on you. And they're relying on you to understand that you're delivering something not just for here and now, but for the future. And you can understand your mortality and lay a future for other growers by developing important germplasm, by introducing traits that are going to allow future breeders to utilize that material to overcome stresses that are forthcoming, and also by developing data that can live beyond your student career. So we're really appreciative of the support we've received. Growers and individuals have donated seeds, libraries. We got Barry Como sent in his graduate advisor's dissertation, photocopies. There's a lot of people that have come out to support us, and we're really excited to start our progress, trying to solve everything everywhere all at once in the realm of grapevine breeding here in Texas.
So most grape breeders I've talked to said it's almost an impossible task to get consumers to adopt new varieties. But then I was shocked to see that you said you're using Munson's varieties as a breeding seed stock because, as I recall, there's not very much vinifera in there. That's mostly exotic stuff.
Yeah, and so one of the biggest obstacles to being a grape breeder anywhere in the world is overcoming the concept that the only useful grapevine is a European grapevine. So Vitis vinifera is the species of European grapevine. It evolved from Vitis vinifera, subspecies sylvestris, diversified, and gained all the domestication traits that we love. Big clusters, hermaphroditic flowers, appropriate chemistry for wine production. We're utilizing Munson material for a number of different reasons. One, because it's persisted in the Texas environment for an extremely long time at this time point in the face of not only those diseases I talked about, but also viral and other challenges. It has a varying level of vinifera from parent to parent. He was utilizing a very diverse base, and one of the cool things about a lot of the private breeders who have popped up over time is that their goals were oftentimes table grapes rather than wine grapes. And so some of his material has really unique and interesting flavors from Vitis labrusca, which is oftentimes overlooked, but has innovated and led to the sort of revolution in table grape quality that we have now. So if you're familiar with cotton candy, a lot of its flavor compounds not only come from muscat, but also come from labrusca backgrounds. The interplay of those aromatic compounds from these different backgrounds give for a lot of excitement. More than anything, Munson did before me what I hope to do, which is collect diversity of Vitis across Texas that has adaptation. Some of it is not going to taste great on the F1, the F2, S0, S1. It's going to take many generations sometimes to get it into a format that is going to be directly what a grower is asking for at this time point. But I'm also seeing, as we're teaching our classes, teaching our viticulture students, they're not necessarily thinking the way that I was being trained when I came into school. They want different grapes. They want lower alcohol grapes. They want lower alcohol wines. They want different flavors. They want different experiences. They want to understand where the grape's from. And there's another opportunity for us through storytelling, to reinvent how we look at grapes and what is quality. And the labrusca has this thing for sugar that's low but still technically ripe, which is a really interesting thing that we can progress into if we're looking for low alcohol wines that are shelf stable and other things.
Thanks so much for the talk. I appreciate
appreciate very much the added context that you've added that I'm sure is heightened when you work with a crop that takes as long as it takes for the final products to be there. And we start our papers with the introduction because we stand on the shoulders of giants, and I believe this is an important part to go back to a little bit more fun. The obviously very charismatic species working with grapes, that snip to sip thing seems very catchy. And something I noticed, it looked like there was almost a subjective consumer element to it. And I noticed that there was a node that said music, and there were some other nodes in it. Does that suggest that someone out there is looking at the data to rank variable importance of factors that influence our taste room?
Yeah, so it's a great question. We have our viticulture, which is where I sit, but we also have enology. And sometimes I get to learn more about grapes and wine and the larger scale when I go to different meetings. Some of the research, one of the publications was Taste the Bass. And the frequency of the music at the time at which you are tasting and sensing can have an influence on your perception of a wine. Not only is the music and the ambience and your experience influenced by your auditory, but also your visual. So I could give you a red and a white wine in black glasses, and if you can't see them, sometimes you can't tell that they're different. You can't discriminate the color for certain products based on flavor alone, although the chemical signatures should be distinct and clear. So there's, I might not get there in my lifetime understanding everything that goes into a sensory experience, but everything that goes into a sensory experience is going to influence our perception of what grapevine we should advance. And so preparing our sensory conditions to understand that and have it as standardized as possible is going to be something that we can do to prevent your favorite song having played in your earphones after you had your favorite lunch, and suddenly this is the grape that's the best in the world. So it's a good question. And so we did, Monday and Tuesday this week, launched some rootstock breeding efforts, collecting a wide array of rootstock germplasm. But from my experience working with growers across the US, especially in light of winter damaging events, which come every 3 to 10 years and take you down to the rootstock, it can be beneficial for a lot of our small farmers to have an all-in-one package. And so developing vines that are capable of reestablishing from the root zone or from tissue really low to the ground is, I think, important. We don't have a decision or an option to not breed for above and below here in Texas. Some of our prominent rootstock options are too vigorous. They lead to a scion overcropping, even though they're capable of expanding our environment, and that has negative repercussions on our fruit and other things. And we have needs for new advanced, like nematode resistance to resistance breaking nematodes. We have to breed for everything everywhere all at once. Yep.
So do you think the increasing trend of high night temperature in case of Texas could be one of the constraints for grape vineyards, whatever we have in Texas?
Yes. So diurnal temperature swings are really important to the final chemistry of our wine and how it ripens. And so we have really hot night temperatures, and that leads to malic acid degradation in our fruit, which leads to low acid grapes at harvest. So we're super sweet, super low acid. That's wonderful for any microbe that wants to eat. And so we have some spoilage issues, some degradation of color that comes from that same sort of lack of acid and breakdown of our compounds in our hot temperatures. There's been a couple different approaches. One is trying to avoid it. So by adjusting the phenology when you select for a really late ripening vine, so that you can try to ripen when there's still some diurnal temperature swing. And that's one approach that we'll have to take for different growing environments so that they can ripen when it's really temperature appropriate. The other pathway that we can take is to try to bring in incredibly high acid germplasm so that we can withstand the summer without losing all of our acid. And fortunately, our work in North Dakota gave us face-to-face opportunities with the highest acid germplasm in the world.
Hi there, I'm sure you're almost out of time, but have you considered trying to use an electronic pump for those in order to get over that barrier of having human taste without
The demand for humans to taste the 1,000 wines that will come from our program might not come from me, but from our industry. Well, they'll want to have humans interact with it. But I'm glad that you brought up, I haven't interacted with an electronic tongue, but electronic noses are regularly used in wine profiling as an adjunct to some of the other aromatic, like compound characterization techniques, GCMS and other stuff like that. So I would do anything to have a technical tongue to fight alongside me, because there's, as good as fruit is, there's a lot of less than desirable flavors that we have to select through on our way to delivering a product to farmers. Grapes.